Imagine a world where we could target like specific enzymes, right?
Okay.
And create therapies that provide pain relief,
Yeah.
but without the high, or reduce anxiety without all those unwanted side effects.
Wow, that would be incredible.
That's the promise of understanding cannabinoid metabolizing enzymes, right?
Yeah.
So today we're going deep into that fascinating world of cannabinoid signaling and enzymatic pathways.
Okay.
Now, you've specifically asked us to focus on FAAH and MAGL,
Right.
two key players in this whole process, and how they're being targeted for drug development.
It's a really exciting area of research, and we're really just starting to sort of uncover the potential of these enzymes.
Before we get into the nitty-gritty of those enzymes, though, let's set the stage a little bit, right? Can you give us a quick overview of the endocannabinoid system and why it's so important?
Absolutely. The endocannabinoid system, or ECS for short, it's like a vast communication network within your body. It plays a crucial role in regulating a wide range of physiological processes, everything from mood and pain perception to appetite and memory.
Wow.
And it uses these special molecules called endocannabinoids as its messengers.
So these messengers are kind of like tiny signals zipping around our bodies, keeping everything running smoothly. Is that a good way to think about it?
That's a great way to put it. And what's fascinating is that scientists have only been seriously studying this system for about 60 years.
Really?
Yeah. The very first synthesis of THC, the psychoactive component of cannabis, wasn't reported until 1967.
1967. That feels so recent.
It does, right. It makes you realize how much we're still learning about this incredible system.
Okay, so how do these endocannabinoids actually work? What are they signaling to?
Well, they bind to specific receptors, primarily CB1 and CB2.
Okay.
CB1 receptors are mainly found in the central nervous system,
Okay.
while CB2 receptors are more prevalent in the immune system.
Okay, so these receptors are like the receiving stations then for the endocannabinoid signals.
Exactly. And when an endocannabinoid binds to its receptor, it triggers a cascade of events inside the cell. This is where things get a bit more complex, but I think we can break it down
Okay.
in a way that makes sense.
Challenge accepted. I'm all ears.
All right, so these CB1 and CB2 receptors belong to a family of proteins called G protein-coupled receptors, or GPCRs for short.
Okay.
GPCRs are like the gatekeepers of the cell, controlling how it responds to various signals from the outside world.
Uh-huh. So the binding of an endocannabinoid to a GPCR is like flipping a switch then. That sets off a chain reaction inside the cell.
That's a great analogy, and this chain reaction involves a whole bunch of other proteins and signaling molecules, ultimately leading to changes in the cell's behavior.
Wow. Can you give us an example of what that chain reaction might look like?
Sure. Let's say an endocannabinoid binds to a CB1 receptor on a neuron in your brain.
Yeah.
This binding causes a conformational change in the receptor, which allows it to interact with a specific type of G protein called gaiose.
Whoa. Hold on a second. Conformational change. G protein. I'm starting to feel like I'm back in biochemistry class.
Don't worry, I promise it's not as complicated as it sounds. Think of it this way. The conformational change is like the receptor adjusting its shape to accommodate the endocannabinoid. And the G protein is like a messenger that relays the signal from the receptor to other parts of the cell.
Okay, I think I'm following so far. So what happens next?
Well, the interaction between the receptor and the G protein triggers a swap.
Okay.
Where a molecule called GDP is exchanged for another molecule called GTP.
Okay.
This exchange causes the G protein to split into subunits, which then go on to activate or inhibit other enzymes and signaling pathways within the cell.
So it's like a domino effect with each step leading to the next.
Precisely. And depending on the specific G protein and the downstream pathways involved, this signaling cascade can have a wide range of effects on the cell's activity.
This is pretty mind-blowing. It's amazing to think that all of this is happening at the molecular level just to regulate things like our mood or appetite.
It really is. And this complexity also means there are many opportunities to fine-tune the system for therapeutic purposes.
Okay, that brings us to the stars of the show today, FAAH and MAGL.
Yeah.
You mentioned earlier that these are key players in cannabinoid metabolism. Can you tell us more about what they do?
Absolutely. FAAH and MAGL are enzymes, which means they're proteins that catalyze or speed up chemical reactions in the body. Specifically, they're responsible for breaking down endocannabinoids, essentially terminating their signaling.
They're like the cleanup crew, making sure the signals don't linger for too long.
Exactly. FAAH stands for fatty acid amide hydrolase, and it primarily breaks down anandamide, one of the most well-studied endocannabinoids. MAGL, on the other hand, stands for monoacylglycerol lipase, and it's responsible for breaking down 2-AG, another key endocannabinoid.
So by controlling the levels of these endocannabinoids, FAAH and MAGL are essentially regulating the duration and intensity of their signaling.
You got it. And this regulation is crucial because you don't want those signals hanging around forever.
Right, yeah.
Imagine if your pain relief signal never turned off, or if your appetite signal stayed on high all the time.
Okay, that makes sense. Now I'm starting to see why these enzymes are so important and why they're such attractive targets for drug development.
You're right on track. By inhibiting these enzymes, we can potentially increase the levels of endocannabinoids, leading to enhanced and prolonged signaling. And this has implications for treating a wide range of conditions, from pain and anxiety to even potentially cancer.
This is all incredibly fascinating. Before we dive into the specifics of how scientists are targeting these enzymes, can you tell us a bit more about the specific pathways that are involved in cannabinoid signaling?
Of course. We've already touched on the GPCR activation cascade.
Right.
But there are other important pathways at play as well, such as the MAPK pathways.
MAPK pathways, what are those?
MAPK stands for mitogen-activated protein kinase. These pathways are involved in a wide variety of cellular processes, including cell growth, differentiation, and even cell death. And they're also modulated by cannabinoid signaling.
So they're like another layer of complexity in the ECS.
Exactly. There are three main MEPK pathways, ERK, JNK, and P38. And each of these pathways can be activated or inhibited by cannabinoids, depending on the specific context.
Okay, I'm starting to see that this is a very intricate system with lots of moving parts.
It definitely is, and we're just scratching the surface of what we know about it.
This has been incredibly helpful in setting the stage. Now, I'm even more excited to learn about how scientists are targeting FAAH and MAGL to develop new therapies.
It's a really fascinating area.
Yeah. Okay, so we've laid the groundwork here. We have a pretty good understanding of how the endocannabinoid system works, the roles of FAAH and MAGL, and even touched on these complex signaling pathways like MAPK.
Yeah. I think we've built a solid foundation for diving into the really interesting stuff, drug development.
Exactly. That's what I'm eager to hear about. How are scientists actually targeting FAAH and MAGL to develop new therapies?
Well, the basic idea is that if you can inhibit these enzymes, you can, increase the levels of anandamide and 2-AG in the body, right? And that could lead to a whole host of therapeutic benefits.
So it's like turning up the volume on the endocannabinoid signaling, is that fair to say?
Yeah, that's a great way to think about it. And there are already a number of drugs in development that do just that.
Oh, wow. Can you give us some examples?
Sure. One example is PF04457845.
Okay.
A potent and selective FAAH inhibitor that has shown promise in preclinical studies for treating pain and anxiety.
PF04457845.
Yeah.
I need to write that one down.
Yeah.
But seriously, it's incredible to think that we could be on the verge of treatments that specifically target these tiny enzymes, right? To manipulate the endocannabinoid system.
It is a really exciting time for this area of research. And PF0444578545 is just one example. There are many other FAAH and MAGL inhibitors in various stages of development.
I'm curious though, if we're increasing endocannabinoids throughout the body, wouldn't that cause unwanted side effects?
That's a very important question, and it's something that researchers are very mindful of. You're right. The endocannabinoid system is involved in a wide range of functions, so just broadly increasing endocannabinoids, could lead to unintended consequences.
So how are they addressing that challenge?
Well, one approach is to develop drugs that are specifically taken up by certain cells or tissues, right, thereby targeting the effects to the desired area. Another approach is to use gene therapy to selectively increase or decrease enzyme activity in specific regions.
Gene therapy, that sounds pretty cutting edge.
It is, but it holds tremendous potential for treating a wide range of diseases.
Yeah.
And as we learn more about the roles of FAA and MAGL in specific cell types and tissues, we'll be able to develop even more targeted and precise therapies.
This is all very promising, but let's not forget about potential downsides, right?
Yeah.
Are there any risks associated with targeting these enzymes?
Of course. Like any drug, FAAH and MAGL inhibitors can have side effects. Some of the potential side effects include nausea, dizziness, and changes in appetite.
Isn't that ironic? Trying to like regulate appetite and then potentially causing appetite changes is a side effect.
It is a bit ironic, but it highlights the complexity of the endocannabinoid system. Finding the right balance is key. And it's important to remember that we're still in the early stages of understanding this system. More research is needed to fully unlock its therapeutic potential and to minimize potential risks.
Well, that's a good reminder that scientific progress takes time. Right. But it sounds like the future of cannabinoid-based therapies is incredibly bright.
I agree. As we continue to unravel the intricacies of the endocannabinoid system and the enzymes involved, we'll be able to develop even more targeted and effective treatments for a wide range of conditions.
What are some of the other conditions besides pain and anxiety that might benefit from these therapies?
Well, the endocannabinoid system has been implicated in a wide range of diseases, from cancer and neurodegenerative disorders to cardiovascular disease and even autoimmune conditions.
Wow, so the possibilities are truly vast.
They are, but as I mentioned earlier, it's important to proceed with caution and to conduct rigorous research to ensure the safety and efficacy of these therapies.
Absolutely. So to summarize, we've learned that by targeting enzymes like FAAH and MAGL, we can manipulate the endocannabinoid system to potentially treat a wide array of conditions.
Right.
We've also discussed the potential benefits and risks associated with these therapies, highlighting the need for continued research.
That's a great summary. And I think it's important to emphasize that while we've focused on FAAH and MAGL today, they're just two players in a much larger network of enzymes and signaling molecules involved in the endocannabinoid system.
So it's like a complex web with each thread connected to another.
Exactly. And this intricate web is what makes the ECS so fascinating and so challenging to study. But as we unravel its complexities, we open up a world of possibilities for developing new and innovative therapies.
Okay, I have one final question before we wrap up this deep dive. We've talked about the potential benefits of increasing endocannabinoids through enzyme inhibition, but what about decreasing them? Are there any situations where that might be beneficial?
That's an intriguing question. And in fact, there are some conditions where lowering endocannabinoid levels might be helpful. For instance, some research suggests that blocking CB1 receptors might be beneficial in treating obesity.
Really? So the opposite of the munchies?
You could say that, but it's important to note that research in this area is still ongoing, and more studies are needed to determine the long-term safety and efficacy of this approach. It's a reminder that the ECS is a complex system, and manipulating it can have both positive and negative effects.
Exactly. It's all about finding that right balance.
Well, on that note, I think it's time to wrap up this deep dive.
I agree. It's been a pleasure exploring the world of cannabinoid metabolizing enzymes with you and our listeners.
The pleasure was all mine. And to our listeners, keep those questions coming. The world of science is vast and ever-changing, and there's always something new to discover. Stay curious, stay engaged, and stay tuned for more deep dives into the fascinating world of science. All this talk about the endocannabinoid system and enzymes, it has me thinking about the potential for treating really complex diseases,
It's definitely an exciting area with a lot of potential. We touched on it briefly earlier, but I'm curious to learn more about the role of cannabinoids in cancer treatment.
That's a great topic to explore. There's a growing body of research suggesting that cannabinoids could play a role in fighting cancer.
What's the connection between the endocannabinoid system and cancer?
Well, studies have shown that cannabinoids can actually exert anti-tumor effects through various mechanisms. Like they can induce apoptosis, which is essentially programmed cell death in cancer cells.
Wow.
They can also inhibit cell proliferation, meaning they slow down the rate at which cancer cells divide and multiply.
Yeah.
And some cannabinoids can even reduce angiogenesis.
Angiogenesis, that sounds intense.
It is. Angiogenesis is the formation of new blood vessels.
Right.
And it's essential for tumor growth. You see, tumors need a steady supply of nutrients and oxygen to survive, and they get that through blood vessels. So by inhibiting angiogenesis, we can essentially cut off the fuel supply to the tumor.
So it's like a multi-pronged attack on the tumor, hitting it from different angles.
Exactly. And what's fascinating is that these effects seem to be mediated by both CB1 and CB2 receptors, as well as other receptors like TRPV1.
TRPV1, that rings a bell. Isn't that involved in sensing heat and pain?
You're right. It's the receptor that responds to capsaicin, the compound that gives chili peppers their heat.
Oh, okay.
And it turns out that some cannabinoids can also activate TRPV1, which may contribute to their pain-relieving effects.
So it seems like cannabinoids have a lot of tools in their arsenal for fighting cancer. That's pretty amazing.
It is, but there's still some challenges to overcome. One concern is the psychoactive effects of THC.
Right. That makes sense. I imagine that wouldn't be ideal for cancer patients who are already dealing with so much.
You're absolutely right. That's why researchers are exploring the use of non-psychoactive cannabinoids like CBD.
Okay.
CBD doesn't bind to CB1 receptors in the brain, so it doesn't produce the high associated with THC.
So they're trying to separate the medicinal benefits from the psychoactive effects.
Exactly. And there's also a lot of research into synthetic cannabinoids that are designed to target specific receptors without activating CB1.
That sounds promising.
Yeah.
Are there any examples of cannabinoid-based therapies that are already being used to treat cancer?
There are. One example is nabilone,
Okay.
a synthetic cannabinoid that has been approved for treating nausea and vomiting associated with chemotherapy.
So it's already being used in a clinical setting.
It is. And some studies suggest that nabilone might also have direct anti-tumor effects.
That's encouraging. It seems like we're just starting to scratch the surface of the potential of cannabinoids in cancer treatment.
I agree. It's a very active area of research, and I'm excited to see what the future holds.
Speaking of research,
Yeah.
what about the enzymes we've been discussing, FAAH and MAGL? Do they play a role in cancer treatment as well?
Potentially, yes. Remember, FAH and MAGL regulate the levels of anandamide and 2-AG, which have, shown to have anti-tumor effects.
Right. So by inhibiting these enzymes, we could potentially boost the levels of these beneficial endocannabinoids.
That's the idea. And some preclinical studies have shown promising results. For instance, inhibiting FAH has been shown to reduce tumor growth in mice.
That's incredible. It sounds like we're adding another weapon to the arsenal against cancer.
It's definitely an exciting avenue of research, but it's important to note that this is still an area of active research, and more studies are needed to determine the safety and efficacy of targeting these enzymes in humans.
Right. It's important to remember that science takes time. We can't rush the process.
Absolutely. But the progress being made in cannabinoid research is truly remarkable, and I'm confident that we'll continue to unlock new therapeutic possibilities in the years to come.
I have no doubt about that. So to bring it all back, we've had an incredible journey today, exploring the endocannabinoid system and the crucial roles of FAH and MAGL. We've delved into the complex signaling pathways, the potential for drug development, and even the exciting possibilities for cancer treatment.
It's been a fantastic conversation, and it's amazing to see how much we're learning about this intricate system and its therapeutic potential.
It really is mind-blowing. And to our listeners, I encourage you to keep learning and exploring this fascinating field. There's still so much to discover, and who knows what breakthroughs await us in the future.
Well said. Remember, knowledge is power, especially when it comes to our health. Stay curious and keep asking questions.
Until next time, stay tuned for more deep dives into the world of science.